Picture two people born in the same hospital on the same day. Same country, same economic circumstances, similar genetics in the broad sense. They grow up in the same neighborhood, attend the same schools, eat roughly the same food, breathe the same air. One of them is statistically likely to die approximately 5 years before the other, not because of any specific illness, not because of any identifiable risk factor unique to their life, simply because of a biological characteristic they were born with and had no say in whatsoever. The one who dies earlier is, in almost every country on Earth, almost every historical period on record, and almost every species of mammal studied, the male. The male longevity gap is one of the most consistent and most puzzling patterns in human biology. It shows up in virtually every dataset that has ever been collected on human lifespan. It predates modern medicine, predates industrialization, predates most of the cultural and behavioral differences between men and women that people reflexively reach for as explanations. It appears in other primates. It appears in wild animals. It appears in laboratory conditions where diet, environment, and behavior are controlled. And yet the full explanation for why it exists remains genuinely contested, operating at the intersection of evolutionary biology, genetics, immunology, endocrinology, behavioral science, and sociology in ways that make anyone who claims a single clean answer is either oversimplifying or selling something. The accurate picture is more interesting than any single explanation. It involves chromosomes, hormones, immune systems, evolutionary trade-offs, cellular mechanisms, and yes, behavior. Not as competing explanations, but as overlapping layers of a biological reality that has been true for longer than our species has existed. Here is what is actually happening. Start with the most fundamental biological layer because this is where the story has its deepest roots and where the evidence is most difficult to argue with. The human female has 2 X chromosomes. The human male has 1 X and 1 Y. This chromosomal difference is the origin of biological sex in humans, and it carries a specific asymmetry that has direct consequences for lifespan. The X chromosome is large, gene-rich, and carries roughly 800 protein-coding genes, along with numerous regulatory sequences. Many of these genes have functions in immune response, DNA repair, cellular maintenance, and other processes relevant to longevity. The Y chromosome is smaller and considerably more gene-poor, with the functional genes it carries being primarily related to male sex determination and sperm production. The consequence of having 2 X chromosomes rather than 1 is genetic redundancy. When a gene on 1 X chromosome carries a variant that reduces its function, a mutation, a polymorphism, a functional impairment, the female has a backup on her second X chromosome. The same gene may be functioning normally on the other copy, compensating for the impaired version. This redundancy applies across hundreds of genes involved in immune function, DNA repair, cellular quality control, and other biological processes that influence how quickly the body accumulates damage over time. Males have no such redundancy for X-linked genes. Whatever version of an X-linked gene they inherited from their mother is the version they have. If that version is suboptimal, there is no backup. The male operates with single copies of several hundred genes for which the female has pairs, and the consequences of impaired function in any of those single copies are expressed directly rather than being buffered by a functional backup. This genetic architecture has measurable consequences. X-linked conditions, genetic disorders encoded on the X chromosome, are dramatically more common and more severe in males who cannot compensate for a defective copy. The immune gene redundancy in females appears to contribute to their generally more robust immune responses, their higher production of antibodies following vaccination, and their lower rates of certain infectious diseases. The same immune robustness that benefits female longevity also contributes to their higher rates of autoimmune conditions, in which an overly active immune system attacks the body's own tissues. A trade-off built into the same genetic architecture. The cellular level adds another dimension. Telomeres, the protective caps on the ends of chromosomes that shorten with each cell division, serving as a biological aging clock, appear to be maintained at longer lengths in females across multiple studies. Telomerase, the enzyme that can rebuild telomere length, shows higher activity in female cells. Longer telomeres mean cells can divide more times before reaching the end-stage dysfunction associated with cellular aging, providing a direct mechanism through which female biology builds in greater cellular longevity from the beginning. Mitochondria, the cellular organelles responsible for energy production, add yet another layer. Mitochondria have their own DNA, separate from nuclear DNA, and this mitochondrial DNA is inherited exclusively from the mother. The mitochondrial genome encodes proteins essential for energy production, and mutations in mitochondrial DNA accumulate over time and contribute to age-related decline. Some evolutionary biologists have argued that mitochondrial DNA is under stronger selection in females than in males because it only passes to the next generation through female reproduction. Natural selection can optimize it for female biology, but is essentially blind to how it performs in males. If mitochondrial DNA variants that are neutral or mildly harmful in males but beneficial in females accumulate in populations, males would systematically carry mitochondrial genomes less well optimized for their biology. This hypothesis, sometimes called mother's curse, predicts a specific pattern of mitochondrial influence on male versus female health that has found some empirical support, though the magnitude of its contribution to the longevity gap remains debated. Now introduce hormones, because this is where the biology gets simultaneously more intuitive and more complicated. Testosterone is the dominant male sex hormone, produced primarily in the testes, and responsible for the developmental and physiological characteristics associated with male biology. It is also associated with a range of effects on longevity that are, on net, negative. High testosterone levels correlate with increased cardiovascular risk. Testosterone promotes red blood cell production, increases certain clotting factors, and influences cholesterol profiles in ways that increase the risk of atherosclerosis, the arterial plaque accumulation that underlies most heart attacks and strokes. Cardiovascular disease is the leading cause of death in most countries and kills men at substantially higher rates than women until the female advantage narrows following menopause. An observation that directly implicates the hormonal environment in cardiovascular mortality differences. Testosterone also influences immune function in ways that are, on balance, immunosuppressive. While the relationship is complex and dose-dependent, high testosterone levels are associated with reduced immune response strength, lower antibody production, and altered inflammatory regulation. The immunological advantage females carry partly reflects the absence of high testosterone and the presence of estrogen, which has generally immunostimulatory effects and may contribute to the more robust immune responses documented in females across multiple study types. The immunosuppressive effect of testosterone has an evolutionary logic. In a sexual selection framework, immune function and secondary sexual characteristics both require metabolic resources, and males may have been under selection to direct more resources toward competitive traits like strength, aggression, and signaling at some cost to immune maintenance. The testosterone-immune trade-off is one of the cleaner examples of evolutionary optimization that trades longevity for reproductive competitiveness. The castration data is the most direct evidence for testosterone's role in male longevity. Studies of historical eunuch populations, men castrated before puberty in various historical contexts, found that castrated males lived significantly longer than intact males of comparable social status, in some analyses approaching female lifespan. The pattern held across different historical periods and geographic contexts, and a study of Korean eunuchs in the royal court found an average lifespan of over 17 years longer than non-castrated males of similar background. This is not a subtle statistical signal, it is a large, consistent effect seen in every historical population of castrated men that has been studied demographically. Removing the source of high testosterone dramatically extended male lifespan. The implication that testosterone is doing something physiologically expensive to male longevity is difficult to escape. Estrogen, by contrast, appears to be actively protective in multiple physiological systems. Its cardiovascular effects include favorable influence on cholesterol profiles, vascular function. By the way, if you want to go even deeper into the secret lives of ancient humans, I just launched a full course called The Secret Lives of Ancient Humans. 10 modules, 3+ hours of content covering everything we never talk about on YouTube. What they feared, how they communicated, what hunted them, and much more. Plus an exclusive PDF guide included. Link in the description. See you inside. And inflammatory markers. Its effects on bone maintenance, cognitive function, and the tissue repair are generally positive during the reproductive years. The postmenopausal reduction in estrogen is associated with increased cardiovascular risk, reduced bone density, and other changes that narrow the female health advantage in the later decades of life, consistent with estrogen having been providing protective effects that diminish with its decline. The cellular maintenance level provides another mechanistic layer. Female cells show higher rates of autophagy, the cellular housekeeping process by which damaged proteins and organelles are degraded and recycled, than male cells in multiple study systems. Higher autophagy efficiency means faster clearance of the cellular debris and damaged components that accumulate with aging, and contribute to age-related dysfunction. The maintenance budget at the cellular level appears to be allocated differently by sex, with female biology investing more in the ongoing repairs that slow the accumulation of cellular damage. DNA repair efficiency shows a similar sex-related pattern, with female cells demonstrating higher activity of certain DNA repair pathways. This matters because DNA damage accumulates continuously as a byproduct of normal metabolism, cellular division, and environmental exposures, and the rate at which it is repaired versus allowed to accumulate determines in part how quickly cellular aging progresses. Now reach into evolutionary theory, because the question of why these biological differences exist requires thinking about what selection pressures produced them. The disposable soma theory, proposed by evolutionary biologist Thomas Kirkwood, provides the foundational framework. The theory proposes that organisms face a fundamental allocation trade-off between resources invested in reproduction and resources invested in somatic maintenance, the repair and maintenance of the body's own tissues. Natural selection maximizes reproductive success, not longevity, and organisms are expected to evolve to invest in maintenance only insofar as it improves reproductive outcomes. For males and females, the optimal maintenance investment may differ because the constraints on reproductive success differ by sex. Female reproductive success in mammals is substantially constrained by the metabolic investment in gestation and lactation. A female who dies young leaves offspring whose survival depends on her continued care. Investment in somatic maintenance that extends lifespans sufficiently to see offspring through dependency may have higher fitness returns for females than for males. Male reproductive success in many species is more constrained by competition for mates than by longevity per se. A male who wins competitive interactions and mates early contributes genes to the next generation even if he dies relatively young. Selection may therefore favor male traits that improve competitive success. Strength, aggression, risk-taking, testosterone-mediated characteristics, even at cost to longevity, because the fitness returns from competitive success outweigh the fitness cost of early death more strongly for males than for females. This evolutionary framework predicts exactly what is observed. Males invest less in somatic maintenance, age faster, and die younger than females in many species, particularly those where male reproductive success is strongly competition-dependent. The human longevity gap fits this predicted pattern and is consistent with patterns seen across other mammals where sexual competition among males is pronounced. The oxidative stress dimension adds biochemical specificity to this framework. Mitochondrial energy production generates reactive oxygen species, chemically reactive molecules that damage cellular components including proteins, lipids, and DNA. Antioxidant defenses neutralize these reactive species, but the balance between production and neutralization determines the net rate of oxidative Damage accumulation, which is a primary driver of cellular aging. Male cells show higher rates of reactive oxygen species production than female cells in multiple study systems, potentially reflecting higher metabolic rates associated with greater muscle mass and the energetic costs of male typical physiology. Female cells show higher antioxidant enzyme activity in some studies. The resulting difference in net oxidative stress would translate directly to faster accumulation of cellular damage in males, a biochemical mechanism consistent with the observed lifespan difference. Move to the behavioral layer, because biology does not operate in isolation from behavior, and the behavioral contribution to male mortality is substantial, even if it is not the whole story. Males die from accidents, homicide, suicide, and risk-associated causes at dramatically higher rates than females in virtually every society and every age group. This is not subtle. In the United States, males account for approximately 3/4 of all accidental deaths, roughly 3/4 of all homicide victims, approximately 3/4 of all suicide deaths. The excess male mortality from behavioral causes is large enough that it contributes meaningfully to the overall lifespan gap, even independent of the biological factors. The behavioral patterns underlying these statistics reflect a complex interaction between biology and socialization. Testosterone does influence risk-taking behavior. The relationship is documented in experimental studies where testosterone administration increases risk tolerance, and this biological contribution to male risk behavior operates alongside social and cultural factors that define masculinity in ways that may encourage risk-taking, discourage help-seeking, and reduce the likelihood that males present for medical care early enough for preventive intervention to be effective. Males in most societies are less likely to visit physicians for preventive care, less likely to present early with symptoms that might indicate developing pathology, and less likely to follow prescribed medical regimens consistently. The behavioral pattern of minimizing health concerns and avoiding medical contact has been documented across multiple cultures and contributes to males being diagnosed with serious conditions at later stages and with worse prognoses than females presenting with the same conditions. This healthcare avoidance is not explained by biology alone. Cultural expectations around male stoicism and self-sufficiency in many societies actively discourage men from treating health symptoms as requiring medical attention. The result is a behavioral pattern with real mortality consequences that interacts with biological vulnerability to produce a compounded disadvantage. Smoking and alcohol consumption, historically much more prevalent in males across most societies, though the gap has narrowed in recent decades, have contributed substantially to excess male mortality from cancer, cardiovascular disease, and liver disease. Occupational hazards have historically fallen disproportionately on males, with dangerous industries involving physical risk, toxic exposures, and injury being predominantly male in workforce composition. These behavioral and occupational contributions are real and meaningful, but they sit on top of a biological foundation that produces lifespan differences even in the absence of behavioral divergence. The immune dimension deserves return, because the immune system's contribution to longevity is one of the more recently appreciated aspects of the lifespan gap. Chronic low-grade inflammation, sometimes called inflammaging, is a driver of age-related disease and functional decline across multiple organ systems. The regulation of inflammatory responses differs by sex in ways that give females better control of chronic inflammation in certain contexts, while also making them more prone to acute inflammatory responses that can be protective against infection. Female immune regulation appears to maintain better balance between inflammatory activation and resolution in many study contexts, contributing to lower rates of certain age-related inflammatory conditions that drive mortality in later life. The same immune robustness contributes to females' better outcomes following many types of infection, better response to vaccination, and more effective wound healing in some contexts. The microbiome, the complex community of microorganisms inhabiting the gut and other body surfaces, shows sex differences that are increasingly recognized as relevant to health outcomes. Sex hormones influence microbiome composition, and microbiome composition in turn influences metabolic function, immune regulation, and inflammation in ways that could plausibly contribute to health and lifespan differences. This is an active and developing research area where the full picture is still emerging, but the direction of evidence is consistent with microbiome differences being one contributing mechanism in a multi-layered biological story. The narrowing of the lifespan gap in certain contexts provides informative boundary conditions on the explanations. In countries where smoking rates have equalized between sexes, the lifespan gap has narrowed. In historical populations and countries where female mortality from childbirth was high, the gap was smaller or absent in some age groups. In laboratory animals where behavior and environment are controlled, a biological gap persists, but at a smaller magnitude than in human populations, suggesting that behavioral factors amplify but do not fully account for the biological difference. The Seventh-day Adventist health studies are often cited in this context. Seventh-day Adventist communities in the United States follow lifestyle guidelines that minimize smoking, alcohol consumption, and many of the behavioral risk factors that disproportionately affect males. And in these communities, the male-female lifespan gap is substantially smaller than in the general American population. This observation is consistent with behavioral factors amplifying a smaller underlying biological gap, rather than behavior being the sole driver. The future trajectory of the lifespan gap is being shaped by converging trends that pull in different directions. Behavioral convergence, women smoking more, drinking more, entering dangerous occupations in greater numbers, has contributed to some narrowing of the gap in countries where these behavioral changes have occurred most dramatically. Advances in cardiovascular medicine that have reduced mortality from heart disease have benefited males more than females in some analyses, because males carried higher cardiovascular mortality rates from which the improvements could provide greater absolute reduction. Improvements in safety regulations and occupational health have reduced excess male mortality from workplace hazards. Against these narrowing trends, rising rates of obesity-related metabolic disease, mental health challenges, and social isolation may be affecting males disproportionately in ways that maintain or reverse the narrowing in certain age groups and populations. The most honest current summary of why men have consistently died before women across history and across cultures is that the gap is produced by multiple overlapping mechanisms, none of which is solely sufficient as an explanation, but all of which contribute in the same direction. The chromosomal architecture gives females genetic redundancy for longevity-relevant genes. The hormonal environment gives females immunological and cardiovascular advantages at the cost of autoimmune vulnerability. The cellular maintenance systems run more efficiently in female biology across multiple measured parameters. The evolutionary optimization of somatic investment differs by sex in ways predicted by reproductive biology theory. The behavioral patterns of males in most societies contribute significant additional mortality from accidents, violence, and preventable disease that accumulates on top of the biological foundation. The person born on the same day in the same hospital, who will statistically outlive their male counterpart by approximately 5 years, is not doing anything special to earn this advantage. They are running on chromosomes, hormones, cellular maintenance programs, and evolutionary history that were not designed. They accumulated layer by layer across hundreds of millions of years of mammalian evolution and were never updated with longevity as a primary objective. Understanding why the gap exists is not an argument for fatalism about male health. Many of the behavioral contributions are modifiable. Medical intervention can address many of the biological risk factors. The gap is smaller in populations with equalized behaviors and better healthcare engagement. But the biological foundation is real, it is deep, and it has been operating long before anyone was keeping statistics. The chromosomes knew before we did.